Welcome for the class. So last videos whichever I have sent you, in that we have seen transcription, translation, lap operon concept. Now next, one of the important concept that we are going to talk about in today's video, that is DNA fingerprinting technique, or in other words it is also called as southern blotting technique. So this DNA fingerprinting technique is mainly based on what we call as VNTRs. VNTRs are variable number of tandem repeats. So about 99.9% of human DNA is similar, okay, but remaining whatever what we call as the 0.1% which is there, that is different in with individuals to individual, and that difference is due to this VNTRs, that is variable number of tandem repeats. These variable number of tandem repeats are the repetitive DNA sequences which are present in the DNA, okay, and that repetitive DNA sequences are called as variable number of tandem repeats. So in this DNA fingerprinting technique, what actually happens, that basic idea I will give you. So this DNA fingerprinting technique is based on VNTRs. We have to isolate the VNTRs, or we have to take out the VNTRs, and we have to study the pattern of what we call as these VNTRs, and by studying this pattern or what we call as the repeats, we will come to know the individual's DNA, and we can compare the individual's DNA, and this technique is called as DNA fingerprinting technique. So this technique is having several steps. So one by one we are going to talk about the steps. So first of all is isolation of DNA. What is isolation of DNA? Isolation of DNA means separating the DNA from the sample. The sample may be a blood stain, it may be a hair sample, it may be a semen stain, it may be a saliva, it may be a bone. So from that sample cells, we can separate the DNA by using some enzymes, and by using what we call as centrifugation method, we can separate the DNA from the sample, and that is called as isolation of DNA. Now after separation of DNA, what will happen? Digestion of DNA. What is digestion of DNA? Digestion of DNA is cutting the DNA into fragments. As you know that DNA, the whole DNA you cannot study it, okay, so that's why DNA is to be cut it into fragments, and that fragmenting the DNA or cutting the DNA is called as digestion of DNA. And this digestion of DNA is done with the help of restriction endonucleases. These are the enzymes which will cut the DNA at specific sites, and they are commonly known as molecular scissors. In detail about this restriction endonuclease, we are going to study in the unit biotechnology. Okay, so the whole DNA is divided into fragments now. That is called as digestion of DNA. That is with the help of restriction endonucleases. Next, third step is separation of DNA fragments. So these DNA fragments are to be separated through a process or a technique called gel electrophoresis. The gel electrophoresis in detail I have discussed in the online class. I will give you a brief idea about the gel electrophoresis. Here the gel is prepared with the help of what we call as agar, and the buffer solution is used, and the wells are created in the gel, and the DNA sample, which is fragmented DNA sample, is loaded in the wells, and what, the electric current is used. Okay, and how the DNA fragments are separated? The principle involved here, the DNA fragments or the DNA is carrying a what? Negative charge. So negatively charged DNA will move towards the positively charged anode. When the power supply is on, the what we call as DNA fragments, based on their size, they start separating. They start moving towards the positively charged anode, and that is called as gel electrophoresis. Okay, next, after this, the transferring of DNA fragments. So now whatever the DNA fragments are there, that is to be transferred. How it is to be transferred? With the help of what we call as a synthetic membrane, such as a nitrocellulose or nylon membrane. Okay, and when you are transferring it, this technique is called as a blotting technique or southern blotting method. So southern blotting method, where on the gel the fragments are present, on that gel the weight is kept and nitrocellulose sheet is also kept, and you have to keep it overnight, and after that you will get what? The fragments will attach to the nitrocellulose membrane. Okay, next step. Next step, that is hybridization step. Before hybridization step, that nitrocellulose membrane is what we call as should react with the alkali to convert the double stranded DNA into a single stranded DNAs. Double stranded DNAs are to be converted into single stranded DNAs, and these single stranded DNAs which are present on the nitrocellulose membrane, okay, that is to be treated with the VNTR probes. Okay, that is to be treated with the VNTR probes. And what are probes? Probes are actually single stranded short, what we call as complementary sequences, complementary to what? The VNTRs which are present on the nitrocellulose membrane. That probes are added, and that probes are single stranded. That probes will go and bind to the complementary sequences which are present on the, or complementary single stranded what we call as DNAs which are present on the nitrocellulose membrane. Now this single stranded probe will bind to the what we call as complementary single stranded DNAs which are present on the nitrocellulose membrane, and forms a hybrid what we call as DNA, and that is called as hybridized DNA. That is called as what? Hybridized DNA, and this process is called as hybridization. This process is called as what? Hybridization. Okay, so DNA probe bind with the complementary sequences of the DNA fragments on the membrane to form a hybridized DNA. Next, what will happen? Now, whatever the hybridized DNA are there, okay, that can be detected by autoradiography. Means hybridized DNA are photographed on what we call as X-ray sheets, X-ray film sheets, and on X-ray film sheets these hybridized DNA, or what we call as the VNTR hybridized probes, okay, these probes forms what we call as the hybridized DNA. They are observed in the dark bands, or they are what seen in the dark bands. Okay, and these dark bands will give an particular pattern in a particular what we call as sample of the DNA. Okay, so this is what detected with the help of autoradiography, that is nothing but the X-ray film. Finally, I will conclude with the help of what we call as applications of DNA fingerprinting. So applications of DNA fingerprinting, in that it is a tool for forensic sciences, okay, to solve the paternity, rape and murder cases. Again, it will be what for the diagnosis of genetic diseases, genetic disorders which are there, that can be diagnosed with the help of what we call as DNA fingerprinting technique. To determine the phylogenetic status of animals, so in evolutionary relationship, if you want to find out the evolutionary relationship, that time you can go for the DNA fingerprinting technique. So if suppose the patterns are resembling, so that time you can conclude that yes, this is this species is what related to what we called as that particular sample, and they are closely related species. Okay, or these two closely related species may have a common ancestors. Like that we can study in the evolution. Next again we can use in the what we call as determination or determine population and genetic diversities. So to determine what we call as population genetic diversities, we can use what we call as DNA fingerprinting technique. Okay, students, I hope you have understood the DNA fingerprinting technique. Once again, I will brief you. DNA fingerprinting technique, which uses the steps as follows. That is, first of all, isolation of DNA, then fragmentation of DNA, then separation of DNA by gel electrophoresis, then sudden what we call as blotting, that the fragments are transferred to the nitrocellulose membrane. Nitrocellulose membrane again treated with the alkali, then it can be transferred to the single stranded DNA. Single stranded DNA are what we call as hybridized with the help of what we call as VNTR probes, and then they are visualized with the autoradiography, that is nothing but the X-ray film. Okay, so students, if any doubt you can discuss in the online classes, okay, or you can personally ask me if you are having any doubt. So thank you, stay home, stay safe.